everyone, welcome back to our channel Ignited Medicos. This is Rachna. In this video, we shall be learning metabolism of cysteine. Cysteine is a sulfur-containing non-essential amino acid which is glucogenic in nature. Non-essential because it can be synthesized from methionine in our body. Methionine is an essential amino acid, so it has to be taken in our diet. Methionine undergoes series of reactions to form cysteine. This in detail is discussed in our previous video, methionine metabolism, so we'll summarize it here. Methionine is catalyzed by methionine adenosyl transferase to form S-adenosyl methionine. This reaction utilizes ATP. Then this is further catalyzed by methyl transferase to form S-adenosyl homocysteine, which further forms homocysteine. Homocysteine condenses with serine to form cystothionein. Cystothionein then forms cysteine. Cysteine is a glucogenic amino acid because it enters citric acid cycle at the level of pyruvate. So in general, amino acids which form intermediates of TCA cycle or enter the cycle at the level of pyruvic acid can form glucose. So they are called glucogenic amino acids. And the amino acids which form acetyl-CoA are ketogenic because they enter TCA cycle, but there is no net synthesis of glucose from acetyl-CoA. Instead, they form ketone bodies, so they are ketogenic amino acid. Some amino acids are both glucogenic and ketogenic in nature, like phenylalanine, tyrosine, isoleucine, and tryptophan. During metabolism, part of carbon skeleton may enter TCA cycle intermediates, whereas other part will generate acetyl-CoA. Like phenylalanine and tyrosine enter TCA cycle at the level of fumarate and isoleucine at succinyl-CoA and tryptophan at the level of pyruvate. These four amino acids are both glucogenic and ketogenic in nature. Leucine is purely ketogenic amino acid, whereas lysine is predominantly ketogenic amino acid in humans. Tyrosine, isoleucine, phenylalanine, tryptophan are both glucogenic and ketogenic amino acids. Remaining 14 amino acids are glucogenic. Here, arginine and histidine are semi-essential amino acids. This is a reversible reaction where 2-cysteine oxidize to form cysteine and cysteine reduce to form cysteine. Now coming to degradation of cysteine. Cysteine on decarboxylation reaction, it forms mercaptoethanolamine, which condenses with pantothenic acid to form coenzyme A. It is important component of fatty acid synthase complex, so it helps in biosynthesis of fatty acids. Next reaction, in the presence of dioxygenase, it forms cysteine sulfite. Again, it undergoes oxidation to form cystic acid, then decarboxylation to form taurine. Taurine condenses with cholic acid to form taurocholic acid, which is a bile salt. Taurine acts as a calcium modulator. It maintains calcium movement. In CNS, it acts as inhibitory neurotransmitter, and it is also important for development of central nervous system, skeletal system, and cardiovascular system. When cysteine sulfite is catalyzed by desulfatase it forms sulfide with the liberation of alanine. This sulfide forms sulfate, which is excreted. But some portion is acted by adenosine to form phosphoadenosine phosphosulfate. This phosphoadenosine phosphosulfate has active sulfur group, which is required for the formation of mucopolysaccharides, and it is also important for detoxification. When cysteine is acted by desulfahydratase, it forms pyruvate with the liberation of hydrogen sulfide and ammonia. Formation of glutathione. Glutathione is abbreviated as GSH to indicate reactive thiol group. Glutathione keeps the enzymes in active state, which is in reduced form. Synthesis of glutathione takes place in two steps. In first step, glutamate combines with cysteine to form gamma-glutamyl cysteine, which is catalyzed by the enzyme gamma-glutamyl cysteine synthase. Then glutathione synthase acts on gamma-glutamyl cysteine to form glutathione. Both the steps need hydrolysis of 1 ATP each. Role of glutathione in absorption of amino acids. For the absorption of amino acids from extracellular space into cell, glutathione is involved in Meister cycle or gamma-glutamyl cycle. It is used for absorption of neutral amino acids. It mainly occurs in small intestine, kidney tubules, and brain. This is extracellular space and this is intracellular space. Here we have phospholipid bilayer, and this is transmembrane protein gamma-glutamyl transpeptidase. Glutathione, which is present inside the cell, binds to a gamma-glutamyl transpeptidase carrier on the inner surface of cell membrane, which breaks down into glutamate, and rest of the molecule will be present inside the cell, whereas glutamate is transferred into extracellular space. This glutamate binds with the amino acid to form gamma-glutamyl amine, which is transported into the cell. Then a free amino acid is released. The remaining molecule which was present in the cell breaks down into glycine and cysteine. And regeneration of glutathione requires 3 ATP. Coenzyme role of glutathione. Glutathione mainly involves in reduction reactions. This hydrogen is used for reducing other substrates like conversion of methyl acetoacetate to tauryl acetoacetate and citric acid to taurine, iodine to hydrogen iodide. Glutathione and RBC integrity. Glutathione is present in RBCs. Glutathione acts as antioxidant, so it inactivates free radicals formed inside RBC. Glutathione is a selenium containing enzyme. Glutathione is regenerated by NADPH dependent glutathione reductase. NADPH is regenerated from glucose 6-phosphate shunt pathway.